The overall goal of this Drosophila brain dissection protocol is to obtain intact brains from adult flies that can be used in a wide range of applications such as immunofluorescence, GFP-tagged protein localization, and ex vivo culturing experiments.
This method can help us answer key questions in the fields of developmental biology and neuroscience, such as the roles of specific proteins and signaling pathways in establishing patterns of neuronal connectivity during brain development.
The main advantage of this technique is that once mastered, it provides a rapid and effective method for immunologically identifying morphological defects in specific regions of the adult brain.
This procedure can be difficult to learn because removing the exoskeleton from the fly head can take a great deal of care and practice. Your movements must be slow and deliberate, and the dissector's arms must be well supported in order to stay steady.
It's a good idea to practice on red-eyed flies and then white-eyed flies before actually dissecting genotypes of interest for actual experiments.
To set up for the dissection, place the anesthetized flies of interest on a cold metal pad or in a petri dish sitting on ice. Alternatively, use a fly pad that emits carbon dioxide. Next, place a small amount of PTN in the center of the dissection dish to create a bubble of PTN. Then, under a stereo microscope, fill the field of view with the PTN bubble under uniform illumination. Now, manipulate flies so that they are belly up and transfer one by the abdomen into the PTN. Completely submerge the animal in the PTN and perform the entire dissection submerged in PTN. With a second pair of forceps, grasp the proboscis.